Hello everyone. Today we have learned about the cell division. There are two kinds of the cell division, that is mitosis and meiosis. So this is my channel Botany Education. You have to watch now. Cell division, as I told you that there are two different kinds of the division. One is mitosis and second one is a meiosis. Mitosis is the first step of the cell division in which the chromosome number is duplicated and equally distributed in the daughter cells. This process of the mitosis takes place in the somatic cells, while meiosis is a second type of cell division. It is also called as the reduction division, in which the chromosome number is reduced and equally distributed to the daughter cells. The process of meiosis takes place in the germ cells, that is in the reproductive cells. Now let us see one by one. These are the different stages of the mitosis and meiosis. So as the parent cell can be divided into the mitotically, so it is having gone through the 4 to 5 stages. The first stage is the interphase, second phase is the prophase, third phase is metaphase, then fourth phase is anaphase, and the fifth phase is the telophase. And here in case of this meiosis, it is a reduction division as shown in this figure. So here the meiosis takes place into two way, that is meiotic division first and meiotic division second. Now let us we focus on the mitosis. So as I just told you that there are the 5 stages of the mitosis. In the interphasic movement, the chromosomes are in a resting condition, and when it is enters into the prophase, after this in the M-phasic movement of the cell cycle, the prophase is just seems like a thread like structure, and they are having the coiling of the chromosome. This is the events of the prophase. While in case of metaphase, it is lie at the equatorial plates, while in case of anaphase, the chromosomes are the repelled from equator and go to the different poles, and in the telophasic movement actual dumbbell shaped structure will be appears. Now let us see one by one. In the interphasic movement, here this is the in that figure as shown in this figure, this is a interphase where the chromosomes are preparing to divide and genetic material to be doubled, and they are in resting phase. Now this is the prophase. The prophase where the, as I just told you, the chromosomes are thread like structure which is thickened, shorter, and become visible to the chromatids joined by the centromeres. Centrioles are moved to the opposite sides of the nucleus. Nucleus will be disappear. Nuclear membrane is also disintegrate. These are the characteristic events of the prophase. Now let us see the metaphase. This is very important stage of the mitosis. This is also where the chromosome is lie at the equatorial plate. The characteristic of this phase is the all chromosomes are come close together at the center and becomes the developing into spindle fibers, centromeres into them, and there are three different kinds of the spindle fibers which is formed, that is continuous spindle fiber, interzonal spindle fiber, and interchromosomal spindle fibers. So this is the characteristic event of this particular phase, in which the chromosomes are the rod shaped, which is lie at the equatorial or middle of the cell, and with the different kinds of the spindle fibers are formed. They get attracted towards the different poles, and with the help of the astral rays, and where the chromosomes their centromeres will be start to divide. Now in next anaphasic movement, where the actual chromosome is pulled apart from one another, say at the middle position where the strength tension will be created, and due to which the centromere will be divides into two, and these two chromatids get attracted toward the different poles, pulling the chromatids toward the opposite poles in this particular phase. So this is where the chromosomes are, their centromere are divided, and they are repelled from the center to the different poles, and this is the characteristic of the anaphasic movement, where the chromosomes are attracted towards the different poles by the due to the disintegration of the spindle fibers, and some of the interchromosomal spindle fibers get attracted towards the pole due to the astral rays and the breaking of the spindle fibers. Now the next phase is the telophase, where the cell division and the division of nucleus will be takes place and also there is a division of the cell can be takes place. That means cytokinesis as well as karyokinesis will be appear over here. The two cell furrows which is go meet at the center, and they forming into dumbbell shaped appearance. The chromosomes are uncoiled, and here the nuclear membrane forms, and the cell will be divides into two. So this is the process known as the mitosis, where the chromosomes and the cells will be divided. Now this is the complete this figure showing the different stages of the complete stages of the mitosis, right from the interphasic movement, prophase, then metaphase, then anaphase, and the telophase. They are occurs in the sequential manner, and after that, this from single cell the two daughter nuclei will be developed, and this is the complete process is known as mitosis, that is also called as the duplication process. Now let us see the next, that is the second cell division, that is stages of meiosis, in which the meiotically it is divided into two. They having two categorized into two stages. One is meiosis first, and the second one is meiosis second. In meiosis first, again they are categorized into four stages: prophase first, metaphase first, anaphase first, telophase first. And in meiosis second, again it is categorized into prophase second, metaphase second, anaphase second, and the telophase second. In the prophase first, where it is divided into again the six substages, that is leptotin, proleptotin, leptotin, zygotin, pacetin, diplotin, and dikinesis. And in metaphase first, again early and the late metaphase first, anaphase first again having early and late, telophase having again early and late. In case of the prophase second, again in meiosis second, these are the simultaneously divides. Now see, this is the graphical representation of the different stages of the meiosis, in which how that chromosomes which is divides and reduct to from the particular parent cell. See here in this particular figure, the prophase first, stages of the metaphase first, anaphase first. In case of the prophase first, that is zygotin is a particular stage in which the synaptonemal complex is developed. In diplotin stage, where the terminalization and formation of the chiasmata, that is in case of the zygotin and diplotin, where the actual process of the exchange of the genetic material can be takes place. So that is a X-shaped chiasmata will be developed in that particular phase. And here the in this after the telophasic first, the vertically it is having the cell furrow, that we called as the septum wall, which is developed, and then later on transversely it will develop and develop that single cell which is reducted into the four different daughter nuclei after the telophasic second. So this is the complete phases of the mitosis and meiosis. This is acknowledgement. I just use some figures from the Google, and that's why I have there written there. So thank you for watching. Thanks a lot.